So I will tell you about the role of fluid flows and how they interact with morphology. And I'm going to look at a specific setup of a morphology, namely at networks. So all organisms of higher life somehow evolved to have vascular or kinds of vascular networks. And I guess the beauty of this network-like structure has, for at least centuries, inspired researchers to think about what drives this architecture. And there are very fruitful approaches to look at these architectures under the umbrella of is the network optimized for, say, minimal dissipation, at keeping sort of the volume cost constant, these kinds of perspectives. And there's beautiful work, and I'm happy to answer any questions regarding the idea of optimizing a network and finding out which kind of network structures are optimal in some regard. However, we are looking here at biological systems. And from the perspective of a biological system, at least for me, it is not clear what the biological system cares about. So what it does optimize for. And in this case, I don't know if you ever, who read Burke-Purcell's Life at Low Reynolds Number? You should all raise your hands next time. It's a night, like you look up his paper and you can read it as a good night story. It's a talk. It's a write-up of a talk. And it has lots of thoughts. It teaches you about life at low Reynolds number, but it has lots of thoughts that sort of go deep also in the meaning of what you're doing when you're doing biological physics. And so one meaning I take when I look at Purcell's Life at Low Reynolds numbers is that he talks about the swimming of E. coli. And he comes to the point that really the beating of the flagella that drives the swimming of this E. coli, it's awfully inefficient. So, did you do the math wrong, and why is it so awfully inefficient? Because he, as a physicist, started with the assumption that this is what E. coli should care for, right? It should care to be a great swimmer, but it does not. And then he even goes to the beautiful argument of saying that, well, maybe what E. coli really cares about is just to go to the next better spot that might have better nutrients. And that's all it's needed. And so as long as the swimming mechanism is good enough to reach that next better spot, it's fine. And it does have lots of other things that the system may care about. And so if we hold on this thought, this is very much when now I look at these vascular networks that I ask, what are the building blocks that drive the systems, and how can I understand what the systems are caring for to understand what drives this network structure? But then again, I'm a physicist. So when I look at these networks now, I want to zoom into a single of these tubes, and I want to look at the role of flows in these tubes. And so the idea that I'd like to teach you today is that the flow in a tube itself is information. Information that can shape the morphology of a network if there's a feedback in response to this information. And here I'm just cartooning out a few ways of how flows in a tube can convey information. So like the fluid flowing through a tube exerts a shear force on the tube wall. That could be rattled. The flow exerts a pressure on the tube wall, or the flow can transport signaling molecules. And now we're looking at the system where these tube walls are not your pipings in your home, but they're actually walls that are alive. So these are the walls of your vascular system, so there your walls align with epithelial cells. And now these walls can respond to this information, and they can self-organize. And so this is intrinsically a physics problem to understand, because flows of fluid in a network are challenging physics. And the reason is pretty simple. The reason is that the flows in a network are all coupled. So if I have a network and I take one of these tubes and I dilate that tube, I not only change the flow in that one tube that I dilated, but I change the flow throughout the network. And so now imagine that I have a tube and it responds to, I don't care, one of these pieces of information, and it dilates. The flow in the other parts of the network changes, and they also respond to that change in information and respond as well. And so you can see how, just based on this principle that flow itself is information and that the tube walls can respond to it, there's a way to self-organize. And I want to teach you a few ideas of how we can go ahead to understand how this feedback may come about. And specifically, this is sort of my own personal working list of things that we should understand regarding the feedback between fluid flows and network morphology. There's flow control, there's adaptability. With adaptability, I coin one term just describing this. If you have a single tube and it's dilating, what does it impact on the global level of the network? There's the role of the topology. So, how big are my loops? How many edges do my loops have? And there's a role of the hierarchy of having bigger veins and smaller veins. And I want to understand how these impact this potential feedback between flow and morphology. And to do that, I will take you on a journey, and I would like to encourage all of you to keep on asking questions. I enjoy that very much. So interrupt me at any time. The roadmap is going to be that I'm going to tell you, first of all, on the question of flow control, or introduce you to a model system where we can aim to understand these sort of dynamics in a more physicsy but bottom-up approach, and then start to progress a tad down that list, and we'll include some like calculations on the blackboard regarding fluid dynamics. I could have also chosen to call this lecture a homage on G.I. Taylor and Taylor dispersion, but I decided to go for flows shaping morphology. But there's going to be a lot of me admiring the works of the old fluid physicists, for example, G.I. Taylor, and why their approach is still very fruitful if we now look at these active systems. So you might have guessed by looking at these networks here that I kind of chose a different network picture than I had on the very first slide, where I showed you the vasculature, or I showed you a fungal network, I showed you a leaf. And now I'm showing you something gray. So that gray is actually not gray. In reality, it's yellow. And it's a slime mold called Phasarum polycephalum that we grow in my lab. So what you see here on the left is a cutout. It's a network. So that's a full organism, Phasarum polycephalum. So it's macroscopic. So that's about like 2 centimeters in size. But it's still a single cell. So it's called a syncytium. Like, for example, you have some neurons that are also syncytia, which means that you have a couple, like in this case, thousands of nuclei, but they're enclosed in a single envelope. Fruit fly embryo is another example of a syncytium. And so what you basically have to imagine now is that what you have here is, in a sense, like a simple cell where you have an actin cortex that is lining the tube wall. This is a zoom in here, and that's surrounding the cytoplasm, and it just happens to be shaped in a network structure.
This is only 2D.
The organism just happily grows in 2D, yeah, on its own. So this is, in this case, it's growing on a petri dish. So it's just a petri dish with some agar. It likes to be in a humid, dark environment, and you happily replenish, provide that in the lab. In the wildlife, it lives in it's a decomposer like fungi. although slime molds are not fungi, but let me just get to that in a bit. So it grows, for example, on wood bark or on leaf litter, and so it eats those and decomposes those. And it also then grows as a 2D network. I mean, if the leaf has a pal, then forms a pal on that one. But yeah, it's basically it grows two-dimensional on its own, which is basically was a high selection criterion to make it onto that slide, and also into my lab, because it's very easy to then follow the dynamics of this network, because you can fully see, you can quantify the full network structure, including the topology, who's connected with whom, which already might be very challenging if you look at a fungal network. And it's not obstructed by any tissue or any other part of the system which you would have in an animal system. Slime molds are not fungi. So the choice of the word mold suggests that they are related. And they are related in their dynamics, in their behavior, in their life cycle, in having such a fungal network. And that sort of gave rise to them being called slime molds. But it doesn't mean that they are the same from the biological makeup. So if you look at the genome of slime molds, of Phasarum, the genome is sort of halfway between fungi and animals. It's actually closer to animals than it is to fungi, but it's basically an old system that branched off in the tree of life early on. And it's actually a key argument for me why it's a good system to look at, because if you were to look at the network dynamics of a fungal network, it looks strikingly similar. So if you have you would, for example, give this organism now some patches of food, and you would see how it reorganizes in response to these patches of food. From the dynamics, it looks strikingly similar to a fungal network. But as I said, the fungal network, it has an entirely different biological makeup. So, I mean, like the fungal hyphae, they're like roots, right? So this is very solid material, whereas here we're talking about like a squishy actin cortex. So it could not be more different, but the dynamics are the same on a coarse-grained level. And that tells me as a physicist that it might not be down to the biological makeup, but it might very much be down to the roles of the fluid flows in these networks that is integrating information, propagating information, that is driving these dynamics. And that sort of justifies why I choose this model system, which is easy to handle in the lab, but otherwise I would not claim that it's of huge ecological importance for the ecosystem of the forest, whereas fungi actually are, by the way, as Agnese nicely pointed out. If you want to build a climate model, your soil is basically fungi, right? This is the key, the dynamics of the fungi. So yeah, going back to the system. So this is the microscopic network, and if we zoom in and we actually look at it under the microscope in life, we see that this actin cortex is undergoing rhythmic contractions, and you can see it nicely if you focus on one of the little bit out of focus veins. You see how they contract and expand. And this is driving the cytoplasm inside to flow back and forth. Oh no, there's a clock running up there. So this is sped up in time a lot. So the typical period of contraction is about 100 seconds. So every 100 seconds, the fluid flows swoosh this way, and and back again. And now, as a first thing to understand, flows in these networks, I want to understand what are the patterns of flows, how they're organized along this macroscopic scales of, say, 2 centimeters. Yeah.
I have a question. I mean, if you say there is this oscillation period of 100 seconds and
yeah.
there is a flow speed, like what fraction of the whole organism can be actively traveled by signals?
Yeah. But that's basically exactly the question I was about to pose. How are the contractions then coordinated across the organism? Like, because that would determine how far something can travel within an allotted time. So is it like random? And so what we actually did is we took this data set that you see looping behind here. And what you can do, which is really basically at the heart of the magic of working with the system, is that you can beautifully quantify these contractions even on this large spatial scale. And the trick behind that is that what you can do is, like, you look at, say, this pixel, and you look at the amount of light transmitted transmitted through the vein at that pixel. And that is really inversely proportional to the diameter of that tube. So you can, at this macroscopic resolution, you can really quantify with very high precision the contractions of every single pixel in this network. And so when doing this, I can now look at these contractions, and I can identify the phase of the contraction. And that's what we get. So this is now really just mapping out the phase of exactly that data set. So the phase goes from black, blue, red, back to black. And you see it really travels along this entire network. It really even matches the wavelengths of this phase, going from 0 to 2 pi, even matches the size of the network. So you have long-range correlations across the network in a single wave. What is even more surprising is not that you only have that at this macroscopic scale of this specimen being 2 centimeters, but the system really adapts to that. So what we did is we took that data, and then we took the specimen out of the microscope, and we cut a piece down here, and we put it back under the microscope. And then we took, again, data, and then we processed the data again. So remember, this is the piece that we cut out. And if we do the same thing to that cut-out piece, it instantly heals. The phase, the contractions reorganize along the new longest axis. And it's roughly still, like if you quantify it, you still find a single wavelength. So going from a wave that was along this direction, in that time that we needed to take it out from the microscope and back in, which is roughly like 5 to 10 minutes, the contractions reorganized along the new longest axis and are still about a wavelength in size. And you can keep on playing this. You can cut it smaller, and you can take lots of different data sets, and you always find this hallmark of a single contraction wave that matches organism size. Yeah.
So inside the tube, cytoplasm?
Yeah.
Outside the tubes?
Air. So air. Yeah. So.
The cell boundary.
So you have you have the cytoplasm, then you have an actin cortex. That's more like a gel-like phase, and then outside the actin cortex you have the membrane, and then outside the membrane you also have some slime, and that's why they're called slime mold. So you have some glycosylix matrix, which is basically their protection shield, and that's it. And then there's air or agar.
There are holes in.
There are holes, yeah. There's nothing in there. So you see, like in the so you might see over the course of, is the movie playing? Oh God, so hard to see. It should be. Oh no, now it's probably playing. Yeah. So this movie is playing, and over the course of, I think the whole movie is like half an hour, you will see veins actually pruning away here. So these shrink away. So the organism is actually actively reorganizing on very long time scales. But for all that matters right now, I will talk on flows and contractions and morphology on a time scale of, say, 10 to 20 minutes, where the network, now you see, like it looks different, right? Where the network topology is very stable. Yeah.
So is this the full cell, or?
Yeah.
And where is the nucleus here?
The nuclei everywhere. So if I were to zoom into one of these bits, and I would do some DAPI staining, I would just in that small, like just the diameter, say a cube of a diameter size. The diameter is around like say 300 micrometers. You would have, I don't know, 1,000, like already. So lots of blue speckles. So there's tons of nuclei there. Yes.
If there is a flow, then how the nucleus are attached to the rings?
What is attached?
There is a flow.
Yeah.
So if the nucleus are inside the flow, they also move with the flow.
Yeah. 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 So, yeah, the flows, which actually, by the way, fungi also do. So, you know, sorry. So also in like fungi, they grow very different. So there you have like single cells, but then they poke holes in the cells to connect them. And also in some, not all fungi, but in some fungi, you have this, that then really the nuclei also travel through several cells. And there's lots of interesting evolutionary aspects about it. So, because how do you mix mutations that come about in this weird networky architecture, and how does it impact whatever? You can see that there's a whole bunch of questions here. What is an interesting bunch of questions here, which I have to admit my lab never ever touched, but it's still intriguing me, is that data from the 60s and 70s suggests that the nuclei divide synchronously. 2 centimeters. I mean, did I mention 2 centimeters? And the nuclei divide synchronously. I don't know. So, and I mean, just to give a scale. So Anne Pringle actually, she once sent me a photo where she was in the woods and she found some phasarum well, some slime mold on a wood bark, and it was seriously that size. Right? They can be much bigger, and synchronous division anyway. So it used to be a model system for cell cycle, because you have this synchronization of the nuclei. Yeah. So there's holes in there. Yeah. And the contractions are coordinated in this pattern now. That did anyone recognize that pattern? This like a sweeping contraction wave. What does it remind you of? You do this when you're swallowing, by the way, for example, all the time. Your intestines do the same thing. Yeah, it's a peristaltic motion. And that actually brings me to, I planned in this wonderful trick, which is, so you have a peristaltic motion. And so we usually only know it in a setting where we have a single tube, right? So we have a contraction that's traveling down. So, and here I show you like the phases of a peristaltic tube. This is along the tube, and this is now over time. You see these traveling waves going through. And that's a tube that's closed. And so the flow goes down at the boundaries. And here in between, you see now in red and blue, that's the flow velocity, red going to one side, blue to the other. And you see the shuttle flow. And the question, well, when you see these flows is now, what does the system care about? And one thought that I'd love you to take away from this lecture is maybe it's transport. And so here I'm mapping out the dispersion of a cloud of particles within that flow field. And it's pretty efficient, and it's probably hopefully answering your question of like how many periods does it take to go from there to like the center that was 1, 2. So in 2 periods you'd spread there. And the key mechanism behind that is the equation that I'm showing up here that's called Taylor dispersion. And this is the equation that I would like to derive with you together right now. Oh, that does not work. So if we think about like this single tube for the moment, what we first going to assume that it's the typical Poiseuille profile that you'll basically always find if your tube diameter is small compared to the length of the tube. So you have then your flow profile is going to be the average flow velocity 1 minus r over a squared. And now we want to understand what's the transport dynamics in these tubes. So we want to solve the diffusion advection equation. And if we write down the diffusion advection equation for the setup, it looks like this. So I have dc dt plus u gradient of the concentration. I only have a flow field along the tube. So that means I have only ur dc dz equal diffusion. And there, all of a sudden now I have the Laplace. So I have dc dz squared plus kappa and the radial component. There's a dr. Sorry. Where did I put it? dc dr. Sorry. So that describes my dynamics of a cloud of particles. And that's a horrible equation, right? Because I have, I got rid of the angular space dimension because the flow profile is symmetric with respect to rotations along the center of the tube. But I still have, I have time, I have space, and I have a radial component. So what you can do is like you can turn on your computer. But turning on your computer is sometimes frustrating because it doesn't tell you which parameters of the system are actually driving a process, right? So that's our desire to find analytical solutions. And G.I. Taylor derived an analytical solution for what you actually care about that I want to show you. And so what you actually care about is not to have the full dynamics of a cloud of particles in that tube. But what I actually care about, if I look at this like on the macroscopic scale on the whole network, I actually only care about knowing the dynamics for the cross-sectional average concentration. So I don't care about the variations along the tube. I only want to see how quickly does it disperse along the tube. And so to derive that, the initial setup is to say that my concentration, I will split it up in two parts, one being the cross-sectional average and one being the variation around it. And I can do the same with my flow field, right? So also my flow field, I can split up in the cross-sectional average and its variation around it, and I actually get that for free the way I wrote down Poisson flow up there. So I know it's U bar, and the variation around it is U bar 1 minus. Oh, sorry. U, I take that out. Oh, there's a 2 here missing. Now it makes much more sense. Okay. And so my aim now is to put in this assumption into this equation and then find an equation for the cross-section average, because this is the only thing that we actually care about. And so if we just plug this in, we find okay, I'm gonna keep this. And then on the right-hand side, or I'll move the right-hand side in, so it's minus kappa . And you see that I already saved one term, which is writing down this expression for the cross-section average, because I know that the cross-section average now only depends on z, right? So this only depends on z and t, whereas this depends on z, r, and t. So I can get rid of this term. Okay, so now I have this equation written out, and now what I do is I take the cross-section average, because that's what I'm actually interested in. I can make use that if I take the cross-section average of the variation, I know that this should be 0, because that's just the definition of how I put it in, that you know, it's the variation around it. And the same is true for this. So when I now take the cross-section average, I find this one's 0, and then plus that's a couple terms, so I don't necessarily know how to equate them. Whereas, oh, sorry, that was wrong. So this term is plus u dc. It's working too far. This I copy, and I just take the average here. This one is a couple terms, so I have no idea how to deal with it. So I'll just write it down that way. This one is the variation. Right here. Goes down to 0 again. And this actually also vanishes, and it's due to the boundary condition that I forgot to state. So the boundary condition is that dc dr at r equal radius equals 0. So there's no flow across the wall boundary. So if I translate this boundary condition, then it tells me from the boundary condition here that dc tilde dr at r equal a should be 0. And you can see that if I take the cross-sectional average, I get, I evaluate at one point this derivative at r equal a, and I also evaluate it at the center. And so I also assume that the solution is symmetric, so there should not be any variational flow across the cross-sectional center. So I also use that dc tilde dr at the center 0 is 0. And then this term vanishes. So that looks pretty good, right? So we have an equation for the cross-section average. So this term's fine, that term's fine. So we're almost there, except for this one, where we have two couple terms and we don't know what happens there. And so now the trick comes in. So now the trick is to try to find an equation for the variation, solve for the variation, and once we have the variation, plug it back in here and equate this term. And then we have the equation for the cross-section average. And so now the trick is to say this is my equation 1, this is my equation 2, and I'm going to do 1 minus 2 to find my the equation for my variation. So now you see why I try to write down this term, and then these cancel. This one goes here. Sorry, need a little more space. And then this one. Okay. Yeah. No. Right. That's not really a nice equation to solve for. So the next trick that you do, which is actually good custom in any case of what you're doing, is always to to simplify, if you end up in with a horrible partial differential equation, to identify what is the order of magnitude of all contributing terms, and which are the dominant terms, and can I solve with just the dominant terms, and then I can try to care if I want to include some of the other terms. So that's a general rule that you should obey to whatever PDE you're looking at. And so there's one, the first assumption that I'm going to use is that in the end I'm interested in the long time solution. So long time solution means that, as I said, I don't care about the variations of my concentration across the mean. So I look at times when the time to diffuse across the cross-section is a squared over kappa. So I look at times when everything has averaged out along the cross-section. And if I do that, then I don't care about this term anymore, because I look at longer times than the time it takes to fuse along the cross-section. And then I also make the statement that if I'm at these long time scales, then the variations should be much smaller than the cross-sectional average. And so now we have, if you look at the dimensions of, say, these four terms for a moment, you have, they all scale with the flow velocity, the length scale along the tube, 1 over L. And so the only thing that's different between among these terms is the cross-sectional variation. And so because I assume that the cross-sectional variation is much smaller than this term, given that all of those are equal by magnitude otherwise, I can neglect these. So that's due to this argument. Maybe I can color code things a little bit. So that one got down with that argument. So the last thing that I'm doing is I'm comparing these two terms, which now scale with diffusivity. One scales, they both have the variation, right? Variation, kappa, same. The only difference is 1 over L squared here and 1 over the tube radius squared here. And since I'm interested in the dynamics of the cross-sectional variation, I assume that the changes along the radius of the tube are much more important than the ones along the tube cross-section, because I'm in the steady state where the cross-section, the variation should not vary much along the tube length. So, and by that argument, I get rid of this term, and then I'm done with, I only have these tubes left as my dominant terms. And these, I'm just copying them down here so that you can appreciate that they are now straightforward to solve.
Can I just
Yeah.
ask a quick question about
Sure.
C tilde being much less than C average? So my impression was that when you run water in a tube,
I know what?
when you run fluid in a tube, like you
Yeah.
have in your figure,
Yeah.
in the middle it's maximum, right? And on the border, on the boundary, it's minimum.
The fluid, yes, the flow velocity. That's this one. That's the Poisson profile. Maximal. This is in the center. It's 2 u bar, and it's 0 here. The flow velocity.
Right. So
Yes.
it's a significant variation.
Yes.
So maybe you can say more about the conditions under which the C tilde is much less than C average holds.
So you can go forward also with the time argument. So what I'm looking at is basically what is the time to diffuse across the tube, which is T diffuse is a squared over kappa, and I compare it to the time to be advected, transported along the tube, and that's SVT L over your average flow velocity. So I compare these two time scales, and I want to be in the regime where the time to diffuse is much faster than the time to be advected. So the correct way of stating this is that this is true. That means that any cross-sectional variations are averaged out by diffusion quickly with respect to the time it takes for like my whatever concentration field to be propagated along the tube. And so you see that basically you have a Peckley number here and A over L there. So it's the Peckley number, which basically is a general number that gives you the time scales between diffusive and effective transport, but also the slenderness of the tube, so the A over L of the tube. So in generally, it always, even if your Peckley number is not perfectly smaller than 1, you can get there by having a long, slender tube, and then this assumption is true. But that's the fixed mathematical formulation for what's blue for this argument. And so now here we arrived at an equation where we know the flow field. It's here. We can plug this in. We just keep this because this does not depend on the radius. And we have just an ordinary differential equation as a function of the radius for the variation of C tilde. And so now we can solve this. I will just for completeness give you the solution so that you have it in case you feel like I would like to go through that exercise. 24a squared. And so since we have this result from this equation, we can now use it and plug it back in here and now solve for the cross-sectional variation. And when we do so, then we arrive at the beloved Taylor dispersion. So and it's really of interesting consequences in now the setting that we're looking at, where we had, well, a network, but let's simplify it to a tube, where we had this tube where we had a shuttle flow. What it meant with the shuttle flow, when you now remember the red and blue going back and forth here, this term is pretty meaningless on the order of a period, because whatever is being transported half a period to the left goes back half a period later. So that's meaningless. So why do we see this strong dispersion up there? What's the role? Well, it's this term. It's this added term in the diffusivity. So kappa is the pure molecular diffusivity. And now we have this additional term which comes in for the cross-sectional dynamics of the concentration along the tube. And it comes in because you have basically diffusion is mixing your particle. You have these streamlines from the Poisson flow. And now your radial diffusion is mixing particles along these streamlines. And so particles hop from fast to slow, from slow to fast, whatsoever. And that spreads it out. And the spreading is proportional to the maximum flow velocity, which basically, given that we have a Poisson profile, gives you the gradient that you have, that the particles can hop along, that they can explore, and that drives the diffusivity. And so you really have, we observe that the system basically does not invest into net transport, because there is no way to have a net transport because you have a closed tube. But it still drives large flow velocities to have this effect of dispersion. And so if you now take this formulation and you put it in a simulation on a network, so this is now just taking the network morphology. There's no data. That's why there's simulation written. So the only data is the network morphology. And what I'm putting down now is a peristaltic motion across that network, and that gives rise to the fluid flows. And I'm solving now the dispersion with the given fluid flows. And then you see how quickly a cloud of particles spreads out along within this network.
Black is the peristaltic motion?
Yeah, kind of, yeah. Yeah, so the black is the network, and I coded in the widths of the segments a little bit, the widths of the radii. Yeah, so you see.
Black is from data?
No, not really. No. So because from data, to solve this accurately, and I did not intend to present it today, but what you have to do is you have to make sure that conservation of mass is fulfilled. And so actually putting down these phases of the contraction, you have to obey conservation of mass. And so that follows some conservation law that I'm obeying here. If you now take the raw data, the raw data, even though we're really, really good at quantifying the contractions, we're not doing like perfect. So conservation of mass is ab initio not fulfilled, but that's not because the system does not fulfill it. It's just because the way we can process the data, or the way we can actually track the dynamics at the end is not perfect. So we would have to add additional measures to make sure that conservation of volume is fulfilled. Otherwise, you only see artifacts because how you calculate flows and networks just goes wrong. But that's why I've just put down a peristaltic wave that obeys this conservation laws. Yes?
Maybe I don't get this whole concept of general dispersion, but what I think is, from what you said, means there's no net flow.
Mm-hmm.
It's only just a dispersion of diffusion.
Yeah, yeah.
But from peristalsis?
Yeah, yeah, peristalsis.
There's a coordinated flow, right? So
yeah.
If there's a contraction, it's from one end, and the flow, that contraction is...
but it flows back again. So it's really important to keep that picture in mind, the one in the center, with the flow velocity. So, and sort of pictured on the network too. So if I'm in the center, I flow blue to the left and red back to the right. So the flow goes forth and back within the single tube.
So what I think,
yeah.
So it's I'm not using your same argument.
Mm-hmm.
This is what is that you have a contraction
mm-hmm.
at one end, what is coordinated, right?
Mm-hmm.
Before you have a distal relaxation of your tube.
Mm-hmm.
Yeah. So if it's coordinated, it should have directionality, because
mm-hmm.
from
yeah.
the image you showed,
yeah, you have a turning wave.
Yeah. The video, actually there was a video that
you
showed this.
It has a direction, yes.
So with that coordinated contraction,
mm-hmm.
there should not be only diffusion. I don't know if...
No, there is, so that's why I'm showing. So there is advection, but it averages out on the time scale of a period. So like this center here more or less stays the center, right? So these particles, they go right and left, but then they go back to the center. They don't travel net. What you have on top of it, which is a little skewing this the dispersion, is that the flow field, this U max in that tube, is not entirely homogeneous because it goes down to 0 at the end. So you have a little less flow velocity here than you have there, and so that's why it's skewed that way. So you see that the maximum kind of shifts a tad like that, but this shift is very, very small. And what's really driving the dynamics is this term, because the diffusivity, if you take, I don't know, glucose, or even if you take a huge protein, for glucose it's 10 to the minus, or like, okay, so we take an ion, so then you have a high diffusivity and it's 10 to minus 10 meters squared per second. And if you take a huge protein, then you are at like 10 to the minus 9. So, I mean, you're basically stuck by the order of magnitude that you can have for diffusion. And so the only way you can outbeat this is having these large-scale flows. Yeah.
Actually, I'm a little confused about sort of the physics of this, you know, effective diffusion constant. So maybe you can say so. So there is kappa, right?
Yeah, this one.
In the absence of flow, and then fluid goes back and forth, right? And then there are proteins in it, right? And somehow, as a result, they diffuse faster proportionally to U squared, which characterizes the fluid.
Yeah.
So, what's the origin of that?
It's
so
the hopping of the.
Like they would go back and then forth.
Yeah.
Back and
So.
forth, and it's average out, but you have U squared, so it doesn't average.
I have this Poisson profile. If I were to be a particle that, say, sits here, and it stays there. Let's imagine it stays here. It goes, in half a period, it goes that way, and the other period, it goes back exactly to that spot.
Yeah, exactly.
But it does diffuse in addition. So while it's going that way, it may travel.
Oh, I see. Okay.
And so it travels to that slower streamline, which means that it doesn't make it back, it only makes it up to here back. And then, so you have this net transport that's proportional to the gradient.
But that's for a particle that starts in the middle. Isn't there the same?
I mean, so yeah. So I mean, that's why we take cross-section average, so that this takes count of all particles starting from any positions, taking into account that if you can hop these lines, that you spread out. So this was just an example like this one. It travels like that, but it doesn't make it back to entirely the spot. But you can also start with one that would travel just a tiny bit back, but then it hops to a faster line and then it gets traveled forward. And so you can see how this is symmetric, because diffusion is symmetric in space. So it spreads out in both ways, because the ones that are slower may get faster on the way back, and the ones that were fast, they get slower on the way back. And so that's why it's symmetric diffusion, and it pops up here.
Very nice. So it's unbiased diffusion.
Yes. Yeah. Yes.
Just a question. Is the movement of the furthest out particle, or so the mean, somehow again diffusive square root in time, or is this something between square root and linear? Or can you quantify the exponent?
The cross-sectional, like, you mean the x squared versus time?
For example, or I don't know, average distance that is traveled.
Yeah, so if you...
Either the boundary of your signal in the subpicture or the center. I mean,
so it's the x squared, like the variation, the variance of that distribution goes with that. The square root of the variance, sorry. The square root of the variance goes with that. Yeah. So there we have the wave and how it's being driven along. And just as a side remark, so one trick I did as a theorist to make this simple is that I took all these tubes to have the same base radius. But it turns out, and the reason why I'm bringing this up, because this is wonderful work from an UNS student who did an internship with me initially when I was at Harvard. So she actually solved this, also taking into account the real hierarchy of the network. And so you have to apologize. I'm going to show you the same data, but she rotated it by 90 degrees. So it looks a little different. But it's the same network. It's just rotated. Sorry for that. So this is the network with without radii, without any hierarchy of bigger and smaller vanes. And this is a network with hierarchy, like the actual hierarchy that we had in the network. And we're now going to follow the spread again. The volume, the total mass is being adjusted, and we followed it for like 250 seconds. And we're going to do the same for the one with hierarchy. Now I just have to pay attention that I stop at the same. Okay, I stopped it a tad earlier. Anyway, you see that it spreads much faster. So the hierarchy plays a role. And we actually found, so that's Sophie that's the student who did this, that if you look at how a network evolves on the timescale of, this is over 3 hours, you see that all these tubes pruned away. And if you were to map out as a measure of how well can it disperse, like an effective dispersion for any point in the network, like if this were my initial point, how quickly could I spread? So basically I showed you one run in the black and copper movie of how a particle would spread. So now imagine I do this for an entire network. This is a map I get for this very interwrapped network. And for the pruned one, you see that there's a lot of variation here, and that the overall dispersion increases by 40%. So by sort of pruning away to few what I call highways, the system really increases the fluid flow along these highways and magnifies the Taylor dispersion effect by 40%. So now I gave you an introduction into the idea of how flows can be controlled in these organisms that I'm looking at, and I used it as to teach you about Taylor dispersion and to make you aware that transport in these fluid networks is of utter importance. But when I started with the question that flows are information. And now that I pitched this, I want to ask which mechanism of information does actually my system of choice use? So how does this organism communicate information? And I know that the system has to communicate information because somehow it's able, on the scale of 2 centimeters, to keep its contraction in phase, right? Because if we have this peristaltic wave on this 2 centimeter scale, somehow this actin myosin cortex has to talk to the other part, actin myosin cortex, like 1.5 centimeters away, to keep them in a checked phase relationship. So how on earth is that possible? And so to identify how, which of these mechanisms of information is being used, Natalie Andrew did the following experiment where she put down a drop of glucose food to the system locally. And what was known is that this drop of food increases the amplitude of the contractions everywhere. So because we put down a local food drop, we know that this increase in amplitude has to propagate in. And so we can identify the velocity with which it propagates in and then delineate. And so this is now taking the data set and just mapping out the increase in amplitude after the stimulus hit. And so increase in amplitude is yellow. You see it starting there, and you see it propagating into the network. And so now you can trust me that I can go in and I can calculate the velocity. When trying to delineate with these different mechanisms of how information can be transported, I was a big fan of elastic waves. So if you go in and you take the physiological parameters of an actin cortex and the cytoplasm and so on, then you'd expect this red shaded area for communication by elastic waves along the system. There was also the idea that you could have, it's a living system, you could have action potentials. And here I'm also mapping out for you velocity of action potentials in light blue. These are all higher organisms with the nervous systems, but also plants, for example, have action potentials, or sponges, and they are a little slower. So these are the dark blue ones. And now we can go in, and now we can measure our front of increased amplitude propagating in. And they are much, much slower. So there's even a gap of 2 orders of magnitude between the next best guess of an action potential. So it's not one of them. That's pretty clear. And when you go actually into the data and get your hands dirty, you see that the stimulus propagation shown in these lines here, these are chymographs. So I took this vein, along the vein, and I folded up in time. The changes between dark and light gray are the contractions. And you see that this is darker here or darker there. So that's the increase in amplitude. And you can measure these velocities there, and they actually agree nicely with the net particle speed. So you have, again, you find that Taylor dispersion is important, that the dispersion of particles initiated at, where you give that food stimulus are propagating information. And so it's, at least on these time scales, it's this mechanism that we're looking at. And so I want you to hold on to this idea that it's, again, the transport that's driving the communication, to now embark on the puzzle of how the hell do the acting contractions, across this 2 centimeters talk to each other? How is it possible that they keep a fixed phase relationship? And that brings us to a theoretical project by another French student. I tend to work with, the initial work was with Gabriel Amsalem, another French, they call it politique these days. So this is the work of Jean-Daniel Juin, who's a wonderful postdoc in the group. And so we embarked on the following. So we said we want to understand how these contraction waves can self-organize. And so we read the literature, and we took into account what's known about an active elastic cortex, like an acting cortex. So we know that this acting cortex, you can have, and in the membrane, you can have channels that are stretch sensitive, and they open up, and then calcium is flowing in, and that calcium can drive the cortex to contract. So we're going to build on this idea that you have a calcium-controlled contractile stress, stretch-activated calcium inflow, and that your actin cortex also has some viscoelastic restoring force. And then so for the calcium dynamics, we can write down the calcium dynamics that's in red. You have like some release due to the stretch of the cortex. That's the cortex R. So that's the stretch of the cortex that gives rise to an increase in calcium, and then you have some sequestration rate afterwards. So that describes the textbook knowledge of how you could describe calcium dynamics. And then we go ahead and we add that the contractions are driving the fluid flows. So if you have now the elastic, viscoelastic restoring stress and the contractile stress acting on the tube wall, it's contracting and it's driving the fluid flow. Again, textbook. And this is just taking account conservation of volume in the tube. So now you can see I can plug this equation in here. So I have a single equation for the radius, and on the previous slide I had an equation for the concentration. And now I'm going to add one single thing. And you can kind of guess what it was. I mean, I designed the whole Taylor dispersion. So now I'm going to add, just as a single added ingredient, that my calcium is also being advected with the fluid flows. So I'm going to make this equation a tad more complicated by adding this term to it. And then I put it on a computer, and I have just two coupled equations. And that makes my life simple, because I can even analytically look for the stability of the system and so on. And I find that indeed, as a function of the parameters of the system, there's a phase where the system undergoes oscillations. And if I go into that phase, I see traveling waves. So this is now, again, taking the geometry of a single tube along the tube over time, mapping out the flow. And this now has a steeper gradient here, but you see, again, forth and back fluid flow. And then one key thing that you should always do when you're a good theorist is you write down equations that are abstracting the dynamics you desire, but you always make sure that you have a best estimate of all your parameters in the system, right? So we go in, and we do the literature search and so on, and we try to give the best estimate for the elastic modulus of the actin cortex, for the viscosity of the cytoplasm, and so on, and so on. And we put them in. We're very proud that we see a traveling wave, but if we put them in, we find that the intrinsic wavelength of our traveling wave is only 7 millimeters. What we observe in the network, but in the system, is that we have traveling waves on the scales of up to 2 centimeters. So there's that difference of like an order of magnitude. So there's not wiggle room enough in the parameters that this might be accounted for that. So something went wrong. But then there's another key insight that what you should do, even if your theory fails, you should still keep looking at it. And so what and JD did this. So he kept looking at it, and he played a little bit with the geometry. And so let me introduce you to the concept what it means to have an intrinsic wavelength. So what it means to have an intrinsic wavelength is you now take a tube and it's closed. Intrinsic wavelength means that if I have a tube that's, say, half the intrinsic wavelength, then the wave that forms, that self-organizes, is going to be exactly the size of the tube, namely half the intrinsic wavelength. So I'm here. And if I make my tube now bigger and bigger, this stays in, they match until I become bigger than the intrinsic wavelengths. And what then happens that you might know from Turing mechanism is that your pattern breaks into part, and all of a sudden you have not a single wavelength, but you have 2 waves and so on. And basically the only thing that you can do is that the wave size that you arrive at are hovering around your intrinsic wavelengths. And there's nothing you can do about it, except for changing the geometry. Because if you look at a loop tube, so periodic boundary conditions, then all of a sudden we do find data points that happily scale along that black line that shows scaling. And the key thing is we can get this reproducibly. We get this reproducibly if you take a tube that's smaller than the intrinsic wavelengths and we slowly grow it. And if we do that, then all of a sudden we can have the wave that forms scale with system size up to about 7 times the intrinsic wavelengths. As a check, I'm also showing you here the geometry of the closed tube. And if we do the same growth procedure with the closed tube, the same observation as before, it scales up to the intrinsic wavelengths, and then the pattern breaks into multiple. And it's only with the periodic or the looped geometry that the system can scale up to 7 times the intrinsic wavelengths, which, given our careful parameter analysis, actually equates to 4.7 centimeters, so well within the arm. So what's going on there? Yes?
When you're saying loop, do you mean that the tube is closed upon
Yeah.
itself? And in what sense is that different from an infinitely sized tube? I mean, if the system is closed upon itself, then the...
It's a periodic boundary condition. I mean, it's not an infinite tube, because even in the infinite tube, like at least on the computer, you have to specify some boundary condition, right? So it's periodic boundary condition, or if you feel like, think about a loop. So what's going on here? So we can show that we can. What's really happening there is that it's the flow at the boundary. Or in this case, the non-existing boundary, but it's the flow that's driving the scaling of this pattern. So the flow that's driving the transport of my signaling molecule, Taylor dispersion, or in this case actually also, is driving the transport. And so the way we can show this is we can take now a tube where we don't put in periodic boundary conditions nor closed boundary conditions, but we put in active flow boundary conditions. So we designate a specific oscillatory flow at one end, and then we check as a function of the baseline and the amplitude of this flow, when do we get scaling? Scaling in this case is yellow. So that was a scenario where we took a a tube that's 5 times the intrinsic wavelength, and you see that basically independent of the net flow velocity, as long as you have an oscillatory component, that drives the scaling. So it's really the flow that drives very unexpected pattern formation in the system. So this concludes my part on flow control, where I introduced you to how flows can scale with network size and how I can get long-range coordination with transport. You have now 2 options, and you can directly choose among them. So one would be, I should manage that. I was wondering, so you probably don't have questions that will last 50 minutes, right? Nope. Okay. So that would give me the opportunity to talk a little bit about adaptability. And the reason why I would care about talking about adaptability is because here I'm going to switch my model system. I'm still going to talk about transport though. So when now turning to adaptability, I want to pose the question, like I have a tube and I dilate it, how does it impact the dynamics on the entire network? And the system now that I want to look at is actually your brain, well, not your brain, but the rat vasculature. And so that's work with Felix Michael. And so this work was very much triggered by this beautiful data set from David Kleinfeld's lab in San Diego. And what they did is they mapped out an excerpt of the rat cortex. So that's the upper part of the cortex, and that's penetrating the nervous tissue, brain tissue. And so what is very remarkable about this data set that it does not only give the vascular architecture, but it also gives the blood flow velocities. So you basically have the full realm of the dynamics of the system at hand, the static dynamics. And now, what is also fascinating about the system, that you do have this effect that individual vessels dilate. So basically what happens if I'm talking to you, or all of a sudden, I don't know, there's a sudden noise in some parts of my nerves, they sort of try to respond to that noise. And so this part of my brain tissue is using more oxygen. And so my neurons then signal to parasite cells that are sort of hugging the vessels in the brain. And they signal to them, I need more oxygen, please relax. And then these parasites relax and dilate the vessel. That can be a very local process. So I have this process of like dilating a vessel, and now I want to understand how much can the supply of oxygen be changed by locally dilating a vessel? I mean, how much do you get out of this? Is this a useful mechanism? And to make progress on that, first of all, again, we need an efficient formula for now absorption. So absorption is the supply of oxygen along the tube vessel. So what we have here, this is basically the same equation as written here. Concentration is diffusing out, being advected by the Poisson profile, and then you have diffusion. And now I'm adding that I also have absorption, so I'm changing my boundary condition here to have now a constant rate of passive uptake along the tube wall. But I can go through the same idea of Taylor by now writing down the concentration of my oxygen or my glucose as a function of the cross-section average and the variation around it, and go through the full spiel again. That's a little more complicated, as you might imagine, and then actually find the steady state absorption along a tube surface. And that I'm going to call phi. And since I'm not showing you the full expression here, you might imagine that it's a little too evolved to show it, so it's still a lengthy expression. But it only depends on basically three non-dimensional parameters and the inflow rate. So the amount of supply you get through that tube, through the wall of a vessel, is proportional to the cross-section of my vessel and the amount of glucose that's flowing in. Then UL over kappa, that is the Packley number, so that gives the timescale of diffusion versus absorption. Then another dimensionless parameter that we identified here, which tells you something about the timescale of absorption versus transport, that we call S. And then this is the non-dimensional uptake rate. So here, this the gamma, there's a gamma. And so now I can look at these dynamics of the system, and I find that they're actually, I can subdivide the parameter space as a function of Packley and S in 3 different regimes. So I find that now this is the timescale of absorption over advection, all in a single tube for now, advection over diffusive transport, the Packley number. I can subdivide the dynamics into 3 regimes. One up here is what we call all regime, where basically whatever comes into a tube, everything is going to be absorbed, of all absorption. One regime that is advection regime, which is given by this formula, which is up here, where transport is mainly advection driven, and one where transport is mainly diffusion driven, that's down here. And now, as I said, there's this wonderful data set by David Kleinfeld's lab. Now we can go in. And because what S and Packley depend on are tube radius, tube lengths, flow velocity in that tube. So we have all these parameters for the red cortex, and so we can just identify where are the vessels in this parameter space. And so what we find is that this advection regime is really dominating for glucose. Just to really make this very, very striking, if you just take an excerpt and you just color in the vessels that are in all the different regimes, like the advective really stands out as being very, very dominant. And so this raised the question, so what is special about this advective transport in being so dominant in the red cortex? Can we explain this from first principles in physics? And so what we now did is we took this data set and we moved our parameters, like the absorption rates, such that we equally cover all 3, all 3 regimes. And now we probe what are the dynamics in these 3 regimes given the real architecture of a red cortex. And so the way we probe this is we take a vessel in each respective regime, we dilate it, and we look at how much is the absorption really going up. So we take a vessel, say, in the diffusive regime, we dilate it by 10%, and we look how much does the absorption go up. So if it goes up by 10%, 0.1, if it goes up by 0.1, so 10%, then you basically get out what you put in. You dilate by 10%, and you get 10% more supply of your glucose. And you see that the response is very different between the regimes. So like in the all regime, it's basically kind of random. It's a very flat distribution, what you get out. Very unpredictable. Whereas the advective regime in dark purple is very, very peaked around that 1.1%, so the 10%, and the purple is doing okay-ish, the diffusive regime. But the advective regime really stands out as being very robust in, even though the flows are globally coupled, the supply is globally coupled, the height breaks loose. If I dilate that one vessel, I still get that 10% increase for a 10% dilation. And the key is we can understand this. I mean, otherwise I wouldn't be talking about that, right? So if we now look at, this was the formula for the supply in the advective regime, and if we now also plug in that the inflow of glucose in that regime is in the advective regime given by this expression, you see that that U cancels out, for example, and that in the end you arrive at an expression that the uptake, the absorption in a vessel in the advective regime is basically given by constants of the vessel. I mean, the constant length, the constant uptake rate, the constant diffusivity, the concentration upstream, and the radius. So I change the radius by 10%. Everything else stays constant. and I get out what I wanted. And so it's very remarkable in that if you have an advective dominated network, then indeed the concentration upstream does stay constant. We can show this. And so you have a very robust increase independent of the hierarchy, the topology whatsoever of the network. And that's also true if you not only look at the single vessel, but you now go in and ask yourself, if I have a structure like, I have a loopy structure, and I say I dilate that vein, how much of, so I know that this goes up by, like if I dilate by 10%, this goes up by 10%, what happens to these and what happens to these? And what these statistics tell you is that these go up and these lose if you are advective or diffusive, if you're all random. So there's a robustness built into, while tuning the radii, overall radii of your vessels to be in this robust regime. So, and again, the key to be able to do this analysis was the simplifications of, that are underlying Taylor dispersion. So with that short story, I gave you an introduction into adaptability, how the increase in transport, flux or absorption can be triggered by local dynamics within the network, even on the global scale. A dream where I can see this going is to think about self-organized optimization of networks because of this global coupling. The role of topology is something we investigate in the group, asking ourselves how can certain topologies stabilize flow pattern, how can, for example, loops in the network increase the overall flow. You've gone on hierarchy. We are very inspired by the idea that you can change the hierarchy by giving specific stimuli to the network. So the hierarchy, like the pattern of bigger and smaller vessels, is actually storing information about the history of events, and you could use this information to, for example, anticipate events. But you're going to learn lots more about this by a beautiful talk by Myrna Krana this afternoon, which is also going to highlight more on the experimental side of our work in the group. And maybe just to conclude, why should we care about networks? I mean, okay, there was the rat cortex, but there was lots of slime molds. Let me pitch that there's a physics of flow networks that needs to be understood, because networks, tubular networks, are everywhere. And even if I'm very selfish and I just look at myself, sure, there's my vasculature, but there's also the porous network in my brain, the cerebral fluid flow. There are my bronchi, which is again the tubular network. There's the nephron network in the kidneys. There are flows in the esophagus and the intestines. There's the lymph system. There's a tumor vascularization. All of these systems are driven by flows in a tubular flow network, and that's not the only ones. As I said, when we're selfish, but if you think about challenges in engineering, tubular networks, like looking at active system, could inspire a self-organization of porous media that are underlying fuel cells or smart materials, or trying to understand synthetic organ printing. So there's a lot to be discovered, and with that, I'd like to thank you as an audience, but thanks also first of all to my team in Göttingen. Thank you.